Hello everyone, my name is Christonia Sproul and I'm a graduate student at North Carolina Central University. Today I would like to share with you a little bit about microarray technology. So what you're going to need in order to follow along is this packet here. This printout here is entitled Microarray. And this is something that I made to help students and to help teachers teach their students about microarray technology. In this packet you'll find background information about microarray and also a link to more information. And you'll also find information about hibernation, which is the condition we're going to use for our experiment today. And you'll also find a link to more information about hibernation. Besides this paper right here, the only thing you'll need is two colors, green and red. It can be crayon, marker, color pencil, whatever you would like. So once you've got your packet and once you've got your colors, the next step would be to divide your classroom into two categories. One category will be hibernation or winter conditions. The other group would be summer conditions. So if you would like to pause right now and divide your class into two separate groups, now would be the perfect time to do that. And join me when you're ready. Alright, so welcome back. Hopefully you've decided what group you're in, summer or winter. Now the next step is, what is microarray technology and how are we going to use it today? So microarray analysis is used in molecular biology and as a diagnostic tool to see what genes are upregulated or downregulated under certain conditions. So if you think about it in terms of puberty, what happens when we hit puberty? Our voice changes in some instances. We get taller. We might have a change in our attitude. Why is this happening? Because the body has triggered these changes because a certain amount of time has passed. So if you have the genes of a student who is not into puberty versus someone who is in puberty, you will see a change. You will see that certain genes will be upregulated, switched on just by the change from being 12 to 13. So microarray technology would allow you to actually analyze those genes to see what genes are actually being turned on when we hit puberty. So that's just one example of how microarray technology works. So here, this is a microarray chip. A typical microarray chip would not be this size. It's only about 3 inches tall. But that 3 inches tall disc could actually hold hundreds of genes, maybe even thousands. So those genes, actually not the RNA of the genes, are put on this chip and analyzed. They are allowed to fluoresce. And based on that fluorescence, microarray technology allows us to see which genes are upregulated and which genes are downregulated. So here is the central dogma. I just wanted to remind you guys of where RNA falls in the central dogma. So as you know, DNA is transcribed into RNA, and that RNA is then translated into proteins. So RNA is what's measured by microarray, because the RNA is the best prediction of what actually will contribute to the function of those genes. Alright, so what is the goal of microarray? There are 3 goals listed here. To find the genes that change expression between experimental designs. You could have a control group that you want to compare to an experimental group. And today, we're going to compare hibernation or winter genes to summer genes. Then microarray will allow us to classify those samples based on their expression profile. And that's what we would see here. The expression profile would show green or red based on whether the gene is upregulated or downregulated. And then from that, we can actually see where patterns occur, or which set of genes are all upregulated together, and we can actually group these genes and say that maybe these genes have the same biological function or contribute to the same biological function. So in order to use microarray technology, in our case, for our experiment, I decided I needed to give you guys a little bit of background about hibernation so that you know what happens to a bear or other animals that hibernate. What happens to them biologically? What's going on in their body? What triggers those changes? And what are those changes? So here I listed just 5 in each category. If you would need more, there are more listed right here in your packet. And for more information, there's actually links for reference for microarray and hibernation in your packet as well. So bears, whenever the winter months start, they're preparing for hibernation, their body temperature begins to lower. They go into this deep sleep. Their heart rate slows. They don't eat, they don't drink, and they don't pass waste during the hibernation months. And all of this is so that the energy is conserved, because it would take energy to eat. It takes energy to keep your body temperature up. So if we can conserve energy, we can keep the bears can keep their body temperatures low for the length of time it's needed in order to survive those harsh conditions. Now, what happens in the summer? We have the temperature normalizing. The bears are definitely more active than they are when they're hibernating. If you think about it, now is the time to hunt. Now is the time to mate. All those things that you won't do while you're hibernating. The heart rate increases. They are eating, and more eating because they need to store fat. That fat will be used while they're hibernating. Typically, bears put on about 5 extra pounds of fat in order to survive hibernation. And also, the energy is used very quickly in the summertime. So now we know the conditions. And again, you can refer to your packet if you need more information. So now we know the conditions that we're going to study. So now let's turn to the 3rd page of your printout. If you have one, great. If you don't, just follow along with the video. On the 3rd page, you'll see the same thing that I have here, right? A list of genes, approximately 24 genes, and your microarray chip. So what I would like for you to do now is take some time within your group and discuss each one of these genes. And decide which genes you feel will be turned on or upregulated under the condition you have been assigned, or which genes will be downregulated under your condition. So I'll do the first one just so you see. The first one says energy storage. So this is gene A. It actually corresponds with the A on your microarray chip. So I would say energy storage would need to be turned on during the winter, because we don't want to burn. Bears would not want to burn energy in the winter. We need to conserve energy, right? So that means I would put a check on the upregulated for energy storage gene, and then I would come over to my microarray chip and color in green where the A gene is, because that gene, in my opinion, would be upregulated. Number 2, or B. Gene B would be increased heart rate. Now, my condition is winter. Your condition may be summer. But in the winter, heart rate, an increase in heart rate, The gene that regulates an increase in heart rate, would it be upregulated or downregulated? I would say downregulated because we need the heart rate to decrease in the winter. So I will put a check in the downregulated. The gene that causes increased heart rate should be downregulated in the winter. So I look for B on my microarray chip and I color it in red. And there you have it. So at this point I would like for you to pause, take the time to actually fill in your table and your microarray chip, and then come back and we'll discuss your results. Now you've taken the time to discuss with your group members which genes are upregulated or downregulated under your conditions. And hopefully you've already taken the time to fill in the check marks accordingly and your microarray chip accordingly. So now I would like for you to compare your results to my results. Once again, the genes that are in green, I felt were upregulated. The genes that are in red are the ones that I feel will be downregulated in the winter. So I have to be specific. Some of you may have summer, some of you may have winter. So these are under winter conditions, hibernating conditions for bears. Now I'll show you the results for the summer. This is how your microarray chip should look for summer. And you notice there's an H for hibernation, right? So if you've done it correctly, you will see an H in your microarray chip. Now what if we compare summer to winter? That's one of the key features of microarrays, to be able to make conclusions based on your results. So here we have winter microarray chip and a summer microarray chip. And as you can see, it looks like the opposite is occurring. Genes that are upregulated in the winter are downregulated in the summer. So microarray technology allows us to get a visual of what's going on the genetic level. So now that we have our conclusion, next we have to discuss what we found. And in the back of your packet you'll find some questions that I've kind of drawn up because I want to make sure that you understand what I wanted you to understand in regards to microarray. So here are the discussion questions. Number 1, in your own words, what is microarray? Now, I'm just going to ask you these questions. Your teacher may decide to pause the video and give you time to answer these questions and then come back. But right now, I'll ask you these questions aloud and you can decide when you want to pause and answer them. So, describe in your own words microarray. The best way to know that if you've learned something is to be able to describe it in your own words. Number 2, what is the significance of the red colors and the green colors? You remember what red stood for versus green? Number 3, what is the main difference that we see between bears' genes in the summer versus bears' genes in the winter? What assumptions can we make based on our microarray results? Number 4, did your microarray match my microarray? If not, give a brief description of the genes that you thought were upregulated or downregulated, and they turned out to be downregulated or upregulated under your conditions. Now this question is going to take some thought because I want you to really analyze the results. And if you have a discrepancy from your microarray and my microarray, it doesn't mean that my microarray has to be correct. Yours could be correct as well. Some genes could be upregulated under both conditions. Some genes could be no change at all. So that's one thing that you may see. In a microarray chip, you would see green, red, and sometimes you see black on a real microarray chip. But this is just to give you the concept of microarray. And number 5, how would you use microarray? So I would like for you to think of your own microarray experiment. Tell me what conditions you're going to test, and then tell me a couple of the genes that you would think would be upregulated and the genes that you would be downregulated. Now this is the fun part. Now you can be creative. Let's say you want to compare, I want to compare my genes to Beyonce's genes. As you can imagine, she probably have upregulated genes related to talent, dancing, singing, that would be downregulated on my side. So, wouldn't we all like to have Beyonce's genes? So be creative and have fun with that one. So I hope you've learned microarray. I hope you feel like you know more about microarray than you did before you watched this video. And I hope I've sparked some type of research, intuition, or some question you'd like to answer in the future using microarray. Good luck, and I'll see you next time.